Welcome back. You've heard all about where we're going in terms of diversity in HCA. Now we have four fantastic speakers to tell you how they're putting all of these principles into practice in their communities. First up is Senjuti Saha from Child Health Research Foundation in Bangladesh.
Hello, everybody. As people start coming in to the room, I am going to tell you a little bit about myself. Pay a lot of attention because this is the most important part of this talk. I am Senjuti, and I do come from Bangladesh. This is a world map. The country you see in red is not Bangladesh. Right, so that's your geography lesson. That's Nepal. This is Bangladesh over here. And you will never forget where Bangladesh is anymore. I come from Bangladesh. I've been working there for about a little over 8 years now. And my team and I are really focused on infectious diseases. So we study bacteria, we study viruses, and so I built the first metagenomics facility in the country, and what we are really interested in is how pathogens spread, antimicrobial resistance, how viruses evolve, brain infections, etc. But it turns out host, which we have been ignoring, but most of the people in this room do not ignore host. Hosts are also important because pathogens and hosts interact, and that is what leads to diseases. And now that we appreciate, as microbiologists more and more, that we need to work with immunologists, we are trying to do that in our organization at the Child Health Research Foundation. And one of the first ways that we are doing that is being part of this pediatric network funded by the Chan Zuckerberg Initiative, and really look at infectious diseases, but through the lens of single-cell genomics of the host. And I call it, the nosedive into leveraging the pediatric network, and I'll tell you more why I call it the nosedive. But really, it's basically a team led by Jose in Boston Children's Hospital. We are 6 different teams located in 5 different countries. And the main objective of this project is not only to do cool science, but also empower, engage, and educate local scientists to be able to contribute to science and answer questions that are relevant to the local populations in these countries. So I'm from Bangladesh. Again, remember where Bangladesh is. And in Bangladesh, we're really interested in viral infections. So here is RSV, also known as respiratory syncytial virus. It's a single-stranded RNA virus. It's the #1 cause of respiratory infections. It causes 33 million cases every year across the world. So it's a very, very democratic virus, and it causes about 100,000 deaths per year. However, when it causes deaths, it's not very democratic. 97% of all mortality due to RSV happens in low-resource settings like Bangladesh, India, and parts of Africa. So there are really many open questions in the field. Why are some children more vulnerable than others? Is there any way that we can predict severity? At least in case of Bangladesh, we want to understand which child is more likely to become severe and perhaps even die from the disease. Can we find some biomarkers that can help us triage patients that are likely to be more severe? And a question we asked is, can single-cell genomics be helpful in our case, right? So in order to at least even answer the question of can we do single-cell genomics in Bangladesh, we designed a study. In this study, we decided to enroll sick children with WHO case definition of respiratory infections. Also, we decided to enroll healthy children because without knowing what healthy looks like, we cannot tell what is different about these sick children. So we decided to enroll healthy controls that are age, sex, and geography matched. We're doing this study in a rural region of Bangladesh called Mirzapur. So we have 5 sites across the country. Mirzapur is our rural site, and we have been working there since 2007. We've been doing a community surveillance there of about 300,000 people who live there. We have a group of 120 health workers who are part of the community, so women and mothers of the community who also have been working with us for many, many years now. There we track every pregnancy, every birth, follow them. Families are visited weekly by these community health workers, and any child who falls sick are referred to our hospital so that we are able to link community data with clinical data and laboratory data. So when we rolled out this study, our study in Bangladesh really started with community engagement. That's how all our community studies start, right? Before we even, design the study, we sit down with our community health workers. We figure out what is most appropriate for the community, what do they want in return, and then only we roll out the study together, right? Community, scientists, hospitals, everybody together. We hired nurses and porters for this study, and they also come from the same community who come to the capital city, work with us for a few days in the lab. They meet the people who are going to work with the samples. They really have ownership of the study. All lab staff also visit the community to see where these samples are really coming from. Meet Muntha. She is our first healthy donor at 8 months and 12 days. So we first got our training in Boston. So this is Dr. Yogesh Huda, a scientist in our lab who went to Boston, worked in Jose's and Alex's lab, tried out a bunch of different technologies, and decided to use honeycomb and bring that technology to Bangladesh, train other scientists in our lab, and also Jose's lab, Faith and Jacqueline, came to Bangladesh, trained us on using the technology and doing some of the bioinformatics work in the country. This study is ongoing. So far, we have collected 147 swabs, 79 that have possible RSV infections or respiratory infections, and the remaining from healthy samples. So far, we are still optimizing. We've only been able to capture cells from 16 swabs and sequence them. So we have about 12,000 cells from these swabs, and we have compared them to about 8,000 healthy children who were enrolled cells from healthy children who were enrolled in Boston. Now, this is very preliminary results, but this is a UMAP that was generated in Bangladesh by Bangladeshi scientists using Bangladeshi samples. The main message that I want you to take away from this is a real UMAP. The cells make sense. We are not seeing cells that we shouldn't be seeing in the nasal mucosa, and very concordant with what we were also seeing from samples collected and sequenced in Boston. The other very interesting thing that we see is when, in these 16 samples, 8 were healthy, 4 were sick, RSV positive, 4 were sick, RSV negative. We do see this, cells that are overrepresented in sick children that are RSV positive. Again, very preliminary results, but these 2 cell types were also elevated in RSV positive cases that were enrolled and sequenced in Boston. Again, gives us hope that, yes, you can do all of this work in Bangladesh and actually apply that for the questions that we are trying to answer. However, it was not an easy journey. The lack of access, we've been talking about it, is really hard to get supplies, and even if you get the supplies, there is no global cost control on reagents, so it's very, very expensive. So while I work in Bangladesh, I pay a lot more than what my friends in the Bay Area of US pay. We have very limited access to compute resources, but thank God for our collaborators who have given us access to Terra. Wasted time when you work with graduate students and takes you 8 months to get simple supplies. It can become very, very hard to continue that work. And because single-cell genomics is still not part of the curriculum, it's really, really hard to retain that expertise because, no matter how many students we train on bioinformatics, everybody wants to go to Canada. So how do we keep that expertise in Bangladesh is also something that we are trying to deal with now. But despite all of these challenges, this is an opportunity like we have had never before. This is an opportunity like no other, right? We are being able to study host and pathogens at the same time. We cannot do cell culture. We do not have fancy facilities to do organoid work. So really, single-cell genomics gives us this way of looking at host and pathogen at the same time in even labs in Bangladesh or other resource-constrained settings. We can contribute to the Human Cell Atlas. And we can talk about diversity, equity, inclusion, and whatever other letters we add to DEI. But for me, for us, it means as Bangladeshi scientists, we have the agency to contribute to global science, working locally. And finally, we had a question about SDG. When we do projects like this, when we look at disease, we look at healthy, we can also contribute in our lifetime to the health and well-being of all. I talked under the umbrella of HCA Asia. I did not talk about Asia. I only talked about Bangladesh and the work we are doing at our foundation. Asia is very, very big. There is no way I will do justice to the amazing work that's happening. In Asia is the largest continent in the world. It has 30% of the land mass, but 60% of the world's population with about 4.7 billion people. So it's really, really important that Asia is represented in the Human Cell Atlas. And there is amazing work happening in HCA Asia. And I invite you to come to our meeting in Hong Kong in December to learn more about the work that's happening in HCA Asia. If you cannot come in person, do join us online. And I'm going to end there saying communities are very, very important to do single-cell genomics. Thank you.
It was inspiring, Senjuti. Thank you. Next speakers, HCA Latin America, Mariana, Boroni, and Patricia, Severino.
Thank you. I'm Patricia. I'm going to be talking to you guys about integrating Latin America to the Human Cell Atlas, and I'm going to be sharing this presentation with my colleague, Mariana Boroni. So just an overview of what Latin America means. We're home to over 660 million people in 33 countries, so that's a lot of diversity. The genetic admixture is really complex. It's involved with our historical colonial past. And the largest population of Afro-descendants outside of Africa is also located in Latin America. It's also home to very diverse groups of indigenous people, so over 800 different groups are located also in Latin America. So we've seen this picture a few times already here in this meeting, and it highlights the diversity mostly of the scientific community engaged with HCA, but not necessarily of the people and the populations that are being profiled. The strategies for expanding Latin American representation in HCA, it's collaboration, obviously, a lot of community engagement and capacity building. And this depends, obviously, on very significant funding. And an opportunity came with the CZI Ancestry Network initiative. And the Ancestry Network call had two important goals, which are related to what we're talking here about, which was to address lack of representation in data generation, but also engaging with communities that are underrepresented in the data set. And a few projects were funded, and these projects are all dealing with Latin American populations and collecting samples from Latin America. Here I highlight two more here. All of them are contributing to the HCA. And I'll be talking specifically about one of them. We call ourselves Latin Cells, so about this one because I'm engaged with this one, but all of them are contributing equally to the HCA purpose. So it's called a Human Cell Map of Latin American Diversity. And this is our team. So our team is composed of 100% Latin people, so from different locations. We're all represented here. And the idea is that we do not all have the same expertise, but we're sharing this expertise among ourselves, and we're all growing together. In the beginning of the project, nobody really had any kind of infrastructure for single-cell analysis, so capacity building was one of the main goals of the project since the beginning, in different levels. Our project has three most important goals. The first one is profiling PBMCs, and why PBMCs? Because those are the most accessible sample types and easy for us to use the same protocols all over the network. We also aim to help creating a new bionetwork for HCA, which is the gallbladder one, and the reason why is because cancer of the gallbladder in Chile is associated with Mapuche ancestry, so this is a very important motivation for the country to be interested in the future to support this kind of network. And of course, the healthy map would be linked with a disease map in the future. And the third one would be to create a Latin American portal for sharing and understanding project outcomes in Latin America. And this portal would be in English, but also in Spanish and Portuguese. As mentioned here before, the language is a barrier, and with this portal, we would be able to talk not only to researchers and students, but also with people who are engaged with the project from the communities to understand the results of the project. I'm going to show you guys one slide per country. I'm starting with Brazil, not because we are Brazilians, but because I put them in alphabetical orders. And I'm not going to tell you everything that is in this slide, but I'm just going to highlight specific points in each one of them. So the first, what I want to highlight about Brazil is very gigantic. And what we aim here is to collect samples from all different locations in this map. So each of the states of Brazil being represented in some ways. Either the participant was born in these regions, or parents and grandparents were born in the region too. We are the hub, one of the hubs for library preparation and sequencing, together with Chile and also Mexico. Here, the Caribbean, we also included the Caribbean in this project. Any of these participants from any of these islands are included at the Mayo Clinic. Hugo here, he's a Mexican colleague of ours, and he recruits people over there. They're very enthusiastic about participating because they really want to contribute to this project, which is so global, and the minorities of their islands would be included. Then Chile. In Chile, they are collecting samples all over the country too. They have ongoing engagement with these communities, so that's why they can collect samples from the indigenous populations at diverse locations. And they also home for our REDCap, so all our metadata is being stored in REDCap and shared among the groups. And it's stored, hosted actually in Chile, right? Colombia. So Colombia is, here I want to highlight in this slide their portable lab. So they have to travel around with a portable lab. The lab is afterwards set on site, like for them to collect samples. They have amazing experiences in the places where they go to, a lot of cultural exchange too. And the idea is that they also have ongoing relationships with these communities for the longest time. That's why they're able to collect samples there. Mexico has a similar scenario. This is their portable lab. The portable lab set there, and they collect samples also all over from communities that they have been engaged with for the longest time. So, and here just to highlight that it's not very easy to get to these places many times. You know, many things happen on the way, and they have to deal with that before they get to the place where they're going to be collecting samples. In Peru, they're targeting one specific ethnic group. They have been also engaged with these communities for the longest time, so they really know how to get around this these locations. And people they always show different interests in why they would participate in the project. Many of them are interested in the ancestry report, but mostly they're very enthusiastic always to represent their peoples in the research project. I brought you this also because there was extra capacity building in Peru. They're like preparing some pisco drinks with some lab parts, you know, and we hope that this was done after sample preparation, but we're not sure. We're going to only know that after we get the results. But this was some extra stuff they did. This is Mexico interacting with Peru here with cultural exchange. And Uruguay does not really have indigenous peoples anymore. They had issues in the 1800s, and they do have a lot of mixed population around the country, and mostly located here. So they also travel here to collect samples in Peru. This is the crew going there. They specifically collect samples at this specific hospital over there. And well, I guess this is my last slide on it, and I would like you guys to have in mind how diverse all these peoples are. Like, all these countries are so different. The cultural aspects of it are very different. The infrastructure is different. So in order for this to happen, it's really complicated. So this is what I wanted to show you guys with these slides, right? We have a final one on the project. So training and research materials are also available because they are part of the project goals, and they involve all the groups working also together. With this, I just want to end with this one about the collaboration that we've been having with HCA since 2019 with the roadshow, and every year we have symposium or workshops which have been supported by the organizing committee, by the researchers from HCA. They're always available to talk to us, always available to talk to students, so it has been really important for us to make all this happen. And I'm gonna now, Mari.
Thank you, Patricia. So while we are building this foundation based on these 4 pillars in Latin America, it's very important to also acknowledge the real changes that we are facing as a community. And thinking on that, 3 years ago, Patricia Posic, who was sitting over there, and we ran a survey in Latin America to kind of understand what are the main challenges and barriers that scientists are facing there. And surprisingly, lack of specialized training came out as the second most important thing or challenge that scientists were facing there. And based on these results, we and others, together with Welcome Connecting Science and Chan Zuckerberg Initiative, we started a task force for capacity building in Latin America. So far, we ran 2 courses, single-cell genomic courses in Latin America, one last year, another one this year. A 3rd course is coming next year. There is also one course that's going to be run in Colombia, led by our colleague Yesid. And also this year, we ran the first symposium on single cell in Latin America. And regarding the course, it was both, the course tackled 2 aspects of single cell. We both trained people how to prepare samples, generate data, and analyze data. We have this, we were worried about teaching using technologies that people has access to kind of overcome the barriers. So we use in-house protocols. Also, Google Colabs, this was led by Vinicius, used to overcome infrastructure barriers. And with those courses, we were able to capacity 4 participants so far from 10 different countries from Latin America. And the course feedback was very positive. And we also thought about how we can keep this community engaged. So after the course, actually during the course, we also ran a grant writing competition, and we support the winners to go to conferences to present their results. We also run some post-course activities like the virtual lab, where PIs are mentoring students all across Latin America to analyze data that is already accessible through public databases. And we are also running monthly webinars where also speakers can show their, not only their work, but also their trajectory. Regarding the symposium, we were able to put together the 2 cohorts of the courses that we gave. Also other participants and researchers from Latin America. And it was a very important place to show their work, to connect, and also to, again, discuss some barriers and challenges that they are facing. And again, we spotted one problem, which is the inequality regarding access to infrastructure. So that's why we are now running another survey to kind of map the landscape of installed capacity in Latin America. So I really would like you guys to please answer the survey and share with your colleagues so then we can have a better picture of what's happening in Latin America. And with that, I really want to acknowledge you all for listening, all the participants from Latin Cells, and all the team that put those courses and symposiums together. Thank you.
Thanks very much, Patricia, Mariana. Great example of community engagement, starting up big things. Next speaker is Kondwani Jambo from the Malawi, Liverpool.
Thank you, everyone, and thank you, the organizers, for inviting me. I'll start with a question. Is there anybody here coming from my continent, Africa? I think that speaks a lot, right? Yeah. Yeah, it speaks a lot about diversity, correct? Yeah, so just like my colleagues that are presented before me, I just want to highlight certain aspects which are important that we should consider going forward. So I'll talk about some facts about Africa. I'll talk about the applications, how we managed to develop single-cell technology in our institute, the challenges we faced, opportunities, and giving you a solution as an example of how we are thinking about this. Maybe some people have seen this map, but it's humbling for me from time to time when I look at it, because the world map as it is drawn is a Mercator map, which means it amplifies certain continents and does not give you a true dimension of how vast the continent of Africa is. It's the second biggest continent and the most diverse. It's 1.5 billion people, about 54 countries. Of course, there should have been more countries. The 54 are just how it was demarcated by colonial people that colonized Africa. To show you that there should be more countries, there are about between 1,000 to 3,000 native languages in Africa. You would find lots of infectious diseases, malaria, TB, HIV, neglected tropical diseases. All these diseases, or exposure to these diseases, or exposure to these pathogens, alter the immune system. So there's a lot of pressure on the immune system. There's diversity from genetics. There's diversity from just the environment. So it's just diversity, diversity, diversity. It's a place rich with diversity. And just to highlight something maybe you might not be aware of, 60% of African population is people under 25 years of age. 42% of the world's youth population in 2030 will be from Africa. That shows you the next 50 years what this could impact, what we study now. I come from the Malawi Liverpool Wellcome Program, which is one of the centers of excellence funded by the Wellcome. There are 5 in the world. One of them happens to be in my country. We have 5 research themes. The important one here for my talk here is the infection biology theme, where the single-cell work sits in. More than 150 active trainees. So we train a lot of people. And we have the know-how of how to build capacity in Africa. We have over 100 active projects, over 20 research groups. Just to highlight that really our mission is to conduct high-quality research to improve health and train the next generation of researchers. And our vision is really science driving health and well-being. I just want to take you through a simple pathway that we took to introduce single-cell technology in Malawi. It's a long journey, lots of failures, but we are now reaping the fruits of persistence. So we started this in 2019 with a collaboration with Alex Chalek, really looking at trying to establish single cell to understand cells from the lung using bronchoalveolar lavage. There were some challenges with this, and in 2020, we managed to get the first 10x Chromium controller and managed to start the single-cell work that we have now and building on that. 2 years later or a year later, we had the Chromium X. So much of the work that we do is either SQL or using the 10X platforms. And we're also, at the moment, working hand-in-hand with Musa, establishing the ATAC-seq technology, and I think that is going well so far. And I'll now take you through, so you can see this has taken about 4 to 5 years. If you did this same thing in the UK or in the US, probably would take you less than 6 months to sort this out. So it took 5 years to get where we are now. The advantage is that we can learn lessons from others. And now we're using this same platform. We're developing our spatial genomics technology. We have been doing spatial biology using microscopy. But now, moving on, we'll build from this. I think it'll be much quicker than the approach we took. But all in all, we've learned a lot of lessons. So these are some of the projects we have at the moment ongoing. They are all to do with infectious disease, one way or the other. So we do studies in, my group does a lot of work on mucosal immunology, mucosal and tissue immunology. We do work around malaria, TB. We are participating in the development of the pediatric single-cell atlas for vaccinated African adults, and all other kinds of studies. But it's just a few of them, just to highlight the diversity of the work that we are involved in. I'll also show you some snippets of some work we have done. And this is our single-cell profiling of alveolar macrophages from healthy individuals. They are healthy because they are not sick, but some of these are HIV infected. So the definition of healthy depends on where you are and the context that you're defining it. So this is work that's done on-site in Malawi, and the component that is not done inside at the moment, which we are planning or working on, is sequencing. But the rest of it, library preparation, preparation of the sample, collecting the sample, all done on-site. This is another project that we're working on, looking at the nasal mucosa. And this uses not swabs, it uses microbiopsies from the nose. And this just shows you an example of the many things that we did. This is comparing HIV-negative individuals, HIV-positive individuals on antiviral therapy 3 months, and those that were more than 1 year on 3 months. You can clearly see, even from the back, that these are different. And mostly what we found is that actually there's an increase in the neutrophil population in the individuals that are more than 1 year on ART. When you look at those that are less than 3 months on ART, there's a lot of antiviral response going on. This is another study that I think was also shown in the last presentations here. Yesterday. This is a spatially resolved single-cell atlas of a fetal COVID in the African population. This was the first study that done in Africa on this. And we're very proud of this work. And this, what it shows is actually that the majority of the cells that were responding in these individuals in the lung were actually macrophages. When you look at other data sets from the world, the majority are neutrophils. So, and this also shown on these images, lots of macrophages responding, and these macrophages were interferon gamma responding macrophages. But when, like I said, you compare to other sites, that's not the case. So just continues to highlight the need to study diverse populations. So in terms of challenges, what are the challenges? They'll be similar to all other sites that I've presented before me. One of them is that it's a longer turnaround time from experimentation itself, sequencing, and data receipt. Meaning if there was a mistake that you made, it takes a long time before you can rectify it. So there is a challenge to that. So that's why it's important to consider all that. The bioinformatic capacity. For example, a computational biologist, that also have a relevant biology expertise. So it's not just being in our case, we need people that are blended, people that can do wet work and people that can do computational biology to understand the process, because we don't have a huge critical mass. But we have been addressing this little by little, and I'll show you how. The limited computational power and data storage capacity, we can underestimate this, but at least now there's cloud storage, so it's improving. And obviously, then the representation of relevant reference data from the African populations to accurately annotate some cell types that probably show up more in our populations than you find elsewhere. The opportunities are there. The opportunities to improvement of discovery research, capacity in LMICs, if we establish this, access to relevant populations with high disease burden, and access to relevant samples from relevant populations. So we are fortunate that we can access these samples, not because people are sick, but for research purposes. We're able to collect tissue samples from people that are healthy in different sites. And as well, most importantly, for the sake of this meeting, contribution to the diversity of the Human Cell Atlas. We are really committed to this goal, and I'm happy to be here. So how are we addressing this? So right now, we are developing this Africa-Asia Single-Cell Genomics Initiative. It involves 5 of the Wellcome-funded programs. So there's one in South Africa, one in Malawi, one in Vietnam, one in Thailand, and one in Kenya. So we've come together. We have diverse areas of expertise. We focus on different diseases, but all that is put together in diseases where they are common in different sites. They work together to address that. And the goal is to establish a network of single-cell genomics hubs to comprehensively profile immune responses to infectious diseases in diverse and underrepresented African and Asian populations. This is being done with collaboration with University of Oxford, the Liverpool School of Tropical Medicine, Wellcome Sanger Institute, Wellcome Connecting Science, and we're working with discussing the Human Cell Atlas to partner on this. But more importantly, I want to highlight this. One of the key things that you see is that when you train computational biologists or people in this kind of expertise, they will leave and go elsewhere. We are working on this philosophy of retain. And this means if you want to retain candidates, you really need to have a competitive remuneration, a culture of excellence, opportunities for training, timely access to resources, impactful research, and a scientific novelty of the work. If we can combine these 6 things, we see that in our setting, we're able to retain people and are able to work on these kind of projects. And lastly, I'd like to thank, this is a huge, it's an opportunity that has been done by a huge number of people. I just want to acknowledge all of them, including the pathologists that are on there that we discussed yesterday, and a lot of other clinicians and collaborators from all over the world. Thank you so much.
Thanks very much, Kondwani, for laying out a lot of the topics we're discussing over here. Last presentation today, Naomi, Habib, and Laleh Haghverdi from HCA Middle East Initiative.
Thanks. And Naomi will be there. Okay. Yeah, hi everyone. Actually, yeah, Naomi and I were supposed to give this talk together. Unfortunately, Naomi could not be here, but she's joining online. Hi, Naomi. You want to say something before I start the presentation? Okay. So we are organizing the Human Cell Middle East community. And yeah, this is more an initiative at the moment. It's not going. Yeah, right. Okay, yes, so here is the advisory team of the Middle East community. It is some people are working in the region, like Yusuf, and Sami, and Naomi, and Luis, and Muhammad, and some of us are also working in Europe or in the US. So today, for example, only Ahmed and I could be here from this whole community. So because of difficulties with the visa, I don't know, other things like the flights being canceled. Anyway, so yeah, that is the Middle East. So I want to say this is the general goals of the Human Cell Atlas, and all of us from also like the Middle East community, we are scientists who contribute to all these general aims and goals, like from spatial data, like Naomi, for example, is doing some work there. Myself, I mean, we have some posters, also some posters from Yusuf's labs. And like, I don't know, foundation models, Mo will present later today. So first of all, so that is the one thing, so that we are really contributing to the whole cause of this Human Cell Atlas effort. in addition to that, we are trying to see like what especially for Middle East can be interesting and how can we contribute from the regional perspective. So this is, of course, characterizing the cell diversity in the population of Middle East. This is maybe more for the scientists who work in the region, so about collecting data, working with patients' material, building biobanks, and data sharing platforms. And then maybe some diseases that are more specific to the Middle East, like so it's not cancer, so maybe some certain types of cancer, but more metabolic diseases and cardiovascular diseases, and also advancing personal and precision medicine. So of course, when you know more detail about the data from that specific region, and creating a knowledge hub and training for the scientists working in the region. So this from the scientific side, but we also want to build a community because, there are certain challenges that we all share. Maybe it is good to talk about them. I don't know, like visa restrictions, travel things. We, of course, want to promote and expose the Middle East scientists' activities and accomplishments. We want to foster regional potentials and networking. So there are some complementary expertise among us. So some countries are more rich with experimental facilities. Some other have more computational people, and we think bringing them together, and if we can manage to make them work together, that would be beneficial for both sides. As I said, we want to see what is the needs in the region and how we can align our research with the needs there, like specific diseases, for example. And also, we want to make a livelihood and good working conditions, good living conditions, like not a lot, but as a community, these are the things that we are interested in. And one important thing is also we are very proud that, there are many Middle East scientists working here despite all the difficulties. We have scientists from Syria, Iraq, so maybe some of you have scientists in your lab from Middle East, or maybe second generations. So we are very proud of that, but this is also something that we want to keep up and keep doing science despite all political instabilities. With that, I want to move to the, one thing, the last time we had, it was in November that we had the virtual conference, the first Human Cell Atlas Middle East Symposium was held virtual. That was a success for us, and just presenting some statistics from that meeting, that it brought about 160 people from 46 countries, 28 speakers, good balance of female and men scientists. And we had both very good science in experiments and computational side. Okay, so sorry for the bad resolution, but that is the map showing the participants. So, so the two biggest hubs are for scientists who are working in the region. Of course, they could bring more from their community, but so we had all these different, also from Iran and India, and we had speakers. It was a very a total enjoyment and pleasure for us how it went. So this was supposed to be our first in-person meeting. So it will be tomorrow morning. We will hold a Middle East session where we discuss some of the challenges and some of the opportunities, like how to get funds together. But yeah, I mean, this is in person, but as I said, because of the travel difficulties, we could not get many people here. So one of the aims is to make a in-person meeting in future in the region. So there are some countries that are more visa-friendly for everyone. And that is one thing that we want to do in the future. We want to also organize workshops and trainings for the students in omics analysis and spatial data analysis. Yeah, tomorrow we will, I said this, and then future goals. Yeah, and we want to also expand a little, like we are very happy to welcome everyone who sees an interest in working with the Middle East community. We were thinking maybe Turkey could also join us. And this is also one of the visa-friendly countries, but let's see. So like whoever sees a link and interest to come and talk to us, just join us tomorrow. That would be great. And we will also discuss what grant application opportunities we get. I think that is it. And Naomi, is there something that you want to add?
Thank you, Laleh. Maybe I'll just say that these are our first steps towards trying and creating a community that would help put kind of the Middle East on the scientific map better. And that despite obvious challenges that we have in the region that are very unique to us, I think the virtual event really showed the potential and interest within the scientific community to promote this single-cell initiative, and we hope for more in-person events in the future.
Thank you. Yeah.
Thank you very much, Laleh. Great talk to round this out. All right, we're going to have a lightning panel discussion now with many of our speakers. Yeah.
I'll give you a mic to share. I like the comment you made about diversity.
All right, there's a lot to go over. Thankfully, we discussed some of these topics in the previous Q&A session with Sophia and Kian Hong and Musa. And our speakers have done an amazing job of going in depth into some of the challenges and opportunities of this kind of research and how you go about it. So I'll try to make this non-redundant because we have very little time, just 24.5 minutes now. I'll request everyone, please keep your questions very short and your answers also as brief as possible. And before getting into some of the topics we've already discussed, I want to bring up the point that, okay, I'll sit down. Bring up the point that there's a lot of exciting science to be done in this area. It's a huge, the fact that it's an untapped field in a sense is exciting. There's a huge amount of interesting questions we can address. And HCA is well poised to do that. So my first question is, what are some of the exciting questions that you feel you can tackle in your work? Kondwani?
So in our case, all the studies we are doing have to be based on a research question. So there are many, like I said, I showed just the flavor of the things we're doing. One of the things I can just point out is about a study that we're working on on the nasal mucosa, and it's not because we just love the nose and it's easy to access. There's a public health need for us to do that. So, for example, there's a bacteria called Streptococcus pneumoniae. It causes pneumonia. It's a huge burden in low-income countries. But one of the things that has been found out is that when people are taking antiviral therapy, they are more likely to carry this bacteria, increasing their chances of getting invasive disease, transmitting in the community, and developing antimicrobial resistance. But we do not know what actually is happening to these individuals at the level of the nose and the level of a single cell. So that's one of the things we're using this opportunity to try to understand that. Thank you.
Okay, fantastic. And on this topic, we mentioned Hari's amazing paper, Nature Medicine, recently on ethnico-ancestral differences in breast tissue. And then I think Kian Hung or Sophia mentioned Jimmy's paper in Science on differences in lupus cellular phenotypes between Asian and European ancestry. And there's a whole slew of single-cell EQTL papers, including Boxiang is in the audience somewhere, just had a paper accepted in Nature Genetics on single-cell splicing EQTLs. This is, again, a major aspect of these cohort studies. Anyone else want to jump in on cool scientific questions you're addressing in this area? Patricia?
Maybe not necessarily a cool scientific question, but the fact that we are working as a network of different countries in Latin America, we are going to try to understand how this ancestry background may affect responses, for example, from the immune system, since we're working with the immune cells. So that would be a first question, and something that has not been necessarily or seen at all from most of the populations that we're going to be including in the project. So it's a very first time that we're going to see what happens.
That's exciting, and certainly the immune system is one of the most rapidly adaptively evolving tissues in the human body because it's constantly fighting with the external world. Yeah, and the external world looks different in different parts of the world. Sorry, go ahead.
Yeah, and on that, we're collecting samples from very different geographical locations, right? So very different backgrounds. So there's a lot of culture and a lot of environment involved. So we do have a lot of variables. We kind of know we're probably going to have something similar to IDA in terms of diversity in our samples. So there's a lot to discuss, I think, until the end of the project related to all those variables that we're including there.
Very much. Senjuti, looks like you want to jump in.
Yeah, so of course I talked about one of the projects that we are doing, but something that I am super excited about, one of the other projects that we are starting, is looking at the impact of preterm birth on immunity and how vaccines affect immunity in children, who were born, or babies who were born preterm and not, because a lot of resource constrained settings has very, very high rates of preterm birth, and we don't really understand if the vaccine dosage, traditional vaccine dosage, work on preterm babies the same way as full-term babies. So that's one of the other studies that we're rolling out, where we are longitudinally enrolling preterm babies, full-term babies, collecting PBMCs, and we'll be following them up for years. And then really working in some of these settings, I think, gives us a lot of opportunities, like looking at the impact of pollution on our nasal mucosa, right? Things that we haven't done a lot of work on. And pollution in different places of Bangladesh is different. So how do different levels of pollution help respiratory diseases, etc.? We're also planning on looking at how the nasal mucosa develops. So when we have community surveillance that has been set up for decades, we can start using our expertise, working with the community, and look at how, for example, the nasal mucosa or other things gradually develop. And these are some of the discussions that we're having with our collaborators.
That's exciting. I think a theme that's coming up from both Patricia and you is the studying the effect of environmental diversity on our biology and how that's important for human health. And it's huge, huge untapped frontier, not just in single-cell research. I'll put in a plug for your amazing cohort, Senjuti, which you've described in various places. If any funders out there want access to the most incredible birth cohort, Dr. Senjuti. Anyone else want to jump in on scientific questions? Maybe bring in Naomi, because she's remote and hasn't spoken yet. Yeah.
Okay, hi. Yeah, I can contribute from our own initiatives related to aging and Alzheimer's disease, where we're now assembling a first initial cohort of a pre-symptomatic early stages of Alzheimer's disease, and a lot of the recommendations, for example, regarding diet, come from the United States, where the initial recommendation is to get a Mediterranean diet. So how does that even apply to our region, where the food is basically a Mediterranean diet, kind of naturally, and yet we still have Alzheimer's in prevalence, and there are a lot of metabolic effects to this and immune effects contributing to healthy aging. So trying to get a local cohort, and also within Israel, we have a diversity. We have different backgrounds and different lifestyles, like comparing people living in the city versus maybe Bedouins or nomads, and all of this is kind of really important because we don't have a good knowledge base even on our own. We need to update it to have a local recommendation for healthy lifestyle that would promote a healthy aging. And it applies to all regions, I would say.
Fantastic. Thanks for bringing up diet, Naomi. Huge impact on our health. And we tried to have some diet metadata in our diversity task force recommendations. Of course, not as deep as someone like you perhaps who's studying it specifically. And we really hope to learn from you the secret of how we can eat Mediterranean food and live very long.
Hopefully.
Sophia?
Sure. So our project came out of observations that we made in the Caribbean with women diagnosed with breast and ovarian cancer and looking at germline genetics. And we saw that there was a variation in the age at diagnosis and also the molecular phenotypes. And of course, in the Caribbean, we're very admixed. We have clearly a large West African population, ancestrally, but also European, Middle Eastern, Asian. And so we wanted to know, based on where we came from predominantly, what was the phenotype of the tumors in other populations. So we expanded our network to include West Africa and East Africa. And while we were doing that, we saw, at least in ovarian cancer, that the types of ovarian cancers that we saw in the West African and East African population was different from the Caribbean Black and US-born Black population in terms of the proportions. And so we're using those questions and these observations to answer questions at the single-cell using the technologies, obviously, for a lot of metadata to ask about exposures, including reproductive factors that also drive disease etiology.
Very, very important question. Really, wherever we look in detail with single-cell analysis, what we're finding is that there's a surprising amount of molecular and cellular differences. And that's exciting because maybe we can finally start to address the differences in disease susceptibility, disease prognosis, drug response, and so on that we're seeing. Actually, personally, to me, it's astounding that the scientific world has waited this long to address all these questions in depth, but that's great. I think the Human Cell Atlas is best positioned to capitalize on that. Yeah. And I think, Laleh, you wanted to jump in as well?
Yes, thank you. For me, it is more computational stuff. Like, of course, we do like data integration, RNA, like cell state velocities, analyzing time series. So these are the problems that are all exciting to us. But I think, like particularly this HCA initiative that we are trying to standardize things, I think that's very exciting. I had a discussion with my lab yesterday that we were comparing with the unit of length and, like how you call it, the kilogram, right? Like everyone was using, you can have different length units, right? So, and then, well, I think that was a very important step to bring it. Like, that's a simple thing, right? So here we have more challenges. It is high dimensional data, many other things. But still, when you think that you are setting up that in future will allow scientists work together and speak the same language, I think that's very exciting.
That's a great new dimension. There are exciting computational challenges as well in this space. Can you highlight one that you're excited about? Something that's related to diversity, which you don't need that algorithm normally for healthy atlases, but if you're studying diversity, you need some kind of methods there.
I mean, there are several things. As I say, like quantifying. I think like understanding the mechanisms is important, right? So like and integrating it maybe with foundation models and. But like really understanding gene networks, how things are going to happen. But this does not mean that other things, trajectory analysis, velocity, they all have their own importance.
Thank you. I think Kondwani, very quickly before we move to the next topic, you wanted to add something?
Yeah, I just wanted to add about one more study that we're working on. My colleague Kayla Barnes is working on. I think most of us maybe think when we develop vaccines, they are tested in high-income countries. They work 90% efficacy. There are a lot of vaccines that are given to infants that are very high efficacy in high-income countries, but the efficacy drops, sometimes even 50% drop in low-income countries. We don't know why that happens, but single-cell technology could be one of the mechanisms we can use to try to understand what is actually happening at a cellular level. And we're actually working on such a project in Malawi. It's also true, I think, in Bangladesh.
That's a great point. Really, the immune system, diversity, and infectious disease comes up a lot, and that does seem to be a priority for this community. Okay, quickly switching gears to another topic. Again, just very brief thoughts, short answers. How does your work benefit, how will your work benefit your local community, and how will it benefit the world? Who wants to start? Short answer. It's a tough question, but you must have said something in your grant proposal.
We do a lot of capacity building from the perspective of training, and it goes beyond the single cell. And that I think is going to go beyond the technology, because we are training physicians on how collaboratively on how to treat women diagnosed with cancer. And it's through that mechanism that we're actually building the single cell atlases. And so for me, that is going to be like long-term impact.
In our case, well, I agree totally with what she said. I think it's totally beyond single cell. But if we stick to the single cell part of it and the molecular biology that we're doing, I think we're supposed to, or we will contribute a lot to how our immune system responds differently to any given situation. I think they highlighted several ones here, and we have the same aim. In our network, people are working with completely different aspects of what we're going to be generating data for. So I think that the contribution is like really huge, because the data that we're going to be generated can be applied to any specific cases that they have locally, and they will have data to compare that with. So I think it's really huge. I think it's hard to imagine how big it is right now, but I think it's going to encompass all these things that have been said about.
Exactly. Even building healthy baselines in different parts of the world, different communities, you'll realize that your criteria of what is normal has to be adjusted, locally. So you've talked about the benefits of essentially the healthy atlasing, but in diverse populations for diagnostics, health monitoring, and so on. Which brings up the question of what is the balance between healthy atlasing and disease applications in your work, again, from a perspective of benefit, right? So anyone else, Naomi, Laleh, Senjuti, Kondwani, you want to talk about that balance and benefit?
Yeah, I think for us, I think it's inherent in the kind of work we do. You always need to have controls. because you can't use controls from a different environment, different setting, different genetic background. So by the nature that we're doing, in our case, we're doing infectious diseases work, there will always be a health control. So I think for us, that's one of the things we think is an easier benefit for contributing to diversity within the Human Cell Atlas. Plus also, I think one of the key things that drives us on the continent is the suboptimal contribution of African science in general in the global science. I think this technology, if democratized properly, has a chance to evolutionize the way we do science on the continent, where there are a lot of the high disease burden and they're contributing to solutions. In this case, I gave an example of trying to understand vaccine responses, why they don't work so well in our setting. There's a direct opportunity there to improve that and increase the effectiveness of these vaccines. Thank you.
That's a fantastic, crisp answer. Thank you so much, Kondwani.
Yeah,
Senjuti?
absolutely. So I'm not going to repeat what he just said, but I want to give a couple of concrete examples. So one of the biggest challenges we had communicating single-cell genomics to the Bangladeshi population is nobody had heard of it. Like students hadn't heard of single-cell genomics. But now, through this project, the moment we generated the first data set, suddenly like journalists got excited and the first UMAP showed up in many newspapers. And now I am inundated with emails. Can we come learn single-cell genomics? I think that's huge. At least people know that term. So that for me is a big reward. The second thing, of course, is through at least the nasal mucosa project, if we are indeed able to find some biomarkers that help us triage patients with most severe cases of RSV, it can immediately lead to policy decisions. So in hospitals during RSV peak seasons, we refuse about 20% of the children because we don't have enough beds. So imagine having a quick biomarker to test patients in the outpatient department and knowing who to admit, right? So that's the second one. And the third one is, all the capacity that we are building, the capacity that we are strengthening, this is just the beginning. As I said during the talk, like single-cell genomics gives us opportunities that we never thought we would have. We cannot do cell culture regularly. We don't have organoids. So now we can start answering questions that are relevant to the local population.
Fantastic. So biomarker discovery for predicting, for prognostics, and for making healthcare more efficient and apportioning resources optimally. Especially for diseases where they're very local. Well, you said RSV is global, but the mortality is very local. So very, very relevant. Naomi, you want to add to this, a balance between healthy atlasing and disease application and how that relates to how your work will benefit the world and the community?
Yeah, thank you. And I really agree with what was said here. I'll just say that from my perspective, we have two kind of parallel efforts that are missing. There's like a need to have better representation of the Middle Eastern population and many other diverse populations in the genetic studies, right? Just the genetic factors, parallel to our efforts of measuring the single-cell profiling. Because at the end, the diversity comes from a combination of the genetics and the environment. And at least in my research, we kept kind of hitting this wall where we don't have enough information and knowledge about our local communities, and it came from both of these axes, actually.
Great comment. Okay, maybe we have very little time left, so I'm going to drop all my questions and move to the audience. I think Moz has a question.
Thank you very much for sharing your experience. It's really amazing, actually, to hear about the journey and the impact it's made. And I guess it's clear that, single-cell atlasing technologies has, really enabled you to ask and move forwards into the research questions alongside the kind of capacity building and strengthening as a result of some of the early grant calls that have been, that have catalyzed this. And what I'm really interested in finding out now is your perspective on how a consortium such as the Human Cell Atlas will be of any value or benefit to moving forwards from where you are now. That would be really interesting to hear your perspectives on.
How can the consortium help you? Great question. Short answers, please. Who wants to start?
I can go.
Senjuti.
More money. But more than more money, really having some influence on cost control. Like, why do we have to pay more in Bangladesh than in San Francisco? It makes no sense. So I think as a consortium, if we can work with all the distributors, all the agents, and ensure that there is equitable pricing, that will make a huge, huge difference.
Thanks, Senjuti. Funding, equitable pricing, very important, high on our agenda. Anyone else want some quick comments on how the HCA can help you? HCA is not a funding agency, but HCA can help work with you to try to organize funding. So very good point. HCA can also work with you to try to get equitable pricing, even though it's not in our control directly, we can lobby. Any other comments on how the HCA can help your work?
Yes, so someone mentioned earlier about advocacy to policymakers. That's really, really important so that folks on the outside can understand beyond our own voice why it's important to do this work and the benefit to the population. So advocacy, not beyond the consumables, but at the government level.
Outreach to government, policy makers, and the community. Great points. Want to bring in more questions? Malta over here.
Yeah, really impressive talks, and the efforts sound really cool. One of the things I noticed is that a lot of you are doing capacity building for training resources, and like retention was mentioned as a challenge. Do you think there is a scope, or is there a need to kind of have a central capacity within the HCA for this, to kind of combine what you already have built and put it in a central area to make sure that's usable also for further expansion? Or also computational resources and stuff like this?
Who wants to go?
Yeah,
Patricia?
the way that do you mean like if you could do something about it, or?
So let's say have a central training portal, which might not be in multiple languages, but something that whatever you're building already could contribute to this and build it, rather than having many different individual kind of local training efforts.
Online resources.
Online resources, for example. Is there? Do you think that's a scope for something?
I think that one of the things that we've seen with what we have been doing during the past years is that language is a barrier. So locally having the courses, and we do have HCA people coming over to participate in the courses. We've done that several times, a lot of support from HCA. So they bring what they know, so it's not separate, it's combined many times with the HCA. I think there are many local ones which we have done that are not directly linked with HCA, but it is somehow, because that's how we started. But the ones that we bring people from HCA and they share, so it's not completely disconnected. And the language barrier is something important. Also, for the symposium, for example, when we're sharing in the discussions, when they can discuss in their local languages, it also helps. I think it makes such a big difference, because many people, they have so much to share.
Language came up before also from HCA Middle East.
Yeah.
Okay, Aviv first and then, Hari first, I think, then Aviv.
Thanks. One of the aspects which the diversity has not covered is the distribution of the adaptive traits in human population, particularly what Kondwani is doing, that their work is certainly influenced by distribution of the Duffy traits, which is more common in the sub-Saharan Africa, which can influence the immune system significantly. So as we move forward, the diversity should consider the adaptive trait distribution. If we consider the Tibetan population, they have a hip to beta mutation, which is embedded in the population. So this is more going back to the scientific aspect of the diversity.
Exactly, and how those adaptive variants can influence your biology in many different ways, as you discussed. We'll take that as a comment and a suggestion rather than a question. Well put. Aviv?
So several of you today, and also, let me do it this way, and also yesterday alluded to the spatial work and to the fact that in many ways it might be faster. For example, the FFPs, I think, were mentioned. And I think we heard that in the center in Malawi, it will be maybe easier to do that. Can you speak a little bit more to what you would need there so that we're not always in catch-up mode, rather than here we have an opportunity to leapfrog and start at the same time rather than later on catch up?
Fantastic. How do we move to the next phase of spatial? And what do you need?
Well, costs for us are almost, well, it's insane how much it costs. So in many of our regions, it's complicated to work with the spatial transcriptomics unless we do have a lot of strong collaborations. And I think strong collaborations maybe are the way to go. Maybe not do it locally even, like maybe do it somewhere else and then send our students there, you know, so they can learn and participate and in a way that maybe we can bring it back afterwards, but maybe buying things locally right now, funding would be the major thing because it's really way more, even more expensive than single cells.
So the opportunity is actually to find these early collaborations so soon that their samples can go somewhere and then come back.
It could be an option, yeah, because,
Just to repeat Habib's point, you're saying that if samples can be shipped to a different place for processing, for example, FFP samples are possible with spatial, that could...
Together with the students and the postdocs.
Exactly.
Their experiment, they just using somebody else's data.
Exactly. Currently, we can ship students, but with spatial, we can ship samples also.
And
yes.
So this is what has happened. Faculty
so
members
just
have
10
come.
seconds.
No, just saying for us, faculty members have come with their blocks from different parts of the world, built the TMAs, we've run the samples, and then they get the data back.
That's a great model.
Yeah, but it costs, like, this is all like crowdsourced funding, so funding for doing that would be awesome.
Thank you so much, Sophia. Great note to end on. Thanks to our panelists once again. Thank you, everyone.